Hello, we are students from Harmony Science Academy, Carrollton.
The principle behind our project was to conduct the procedures of ELISA in correlation to detecting certain hormones within female pandas to determine their periods of ovulation. ELISA stands for enzyme-linked immunosorbent assay and is used to detect antibodies and hormones. The project revolves around reproductive endocrinology, reproductive hormones, and how ELISA can help female pandas ovulate.
For our ELA component, we created a newspaper to highlight the importance of ELISA and how it helps pandas in their population. We created articles and interviews to show the importance of raising the panda population.
For our social studies component, we chose to focus on the historical and economic aspect of ELISA, including the researchers who created ELISA as well as the marketing and diversity in cost and availability. For more information of our ELA and social studies component, refer to our website.
The materials we used were 7 colored tubes, 20 to 200 microliter adjustable micropipette, yellow tips, disposable plastic transfer, 70 to 80 milliliter wash buffer and beaker, paper towels, and gloves.
We labeled one well strip 1 through 12 to act as a standard hormone reference, and another with 3 positive, 3 negative, 3 for panda A, and the remaining 3 for panda B. Going back to the first well strip, 50 microliters of phosphate buffered saline is pipetted into the wells labeled 2 to 12. 100 microliters of the primary antibody is added to the first well, then 50 microliters is consecutively pipetted out of the initial well into the next. The process stopped at the 11th well. The leftover 50 microliters is discarded. Using the second well strip, 50 microliters of the positive and negative control is transferred into the next 6 wells labeled positive and negative. Similarly, 50 microliters of the panda A urine sample and the panda B urine sample is transferred into the next 6 wells labeled panda A and panda B.
Wash buffers pipetted to the tops of the wells in between the periods of which we added the primary and secondary antibodies, which both rested for 5 minutes for binding purposes.
When the substrate is added to the enzyme, the solution changes colors, and in this case it changed to blue. The more blue the solution is, the more hormones are present. In the positive control, the solution was blue to show hormones were present, but in the negative control, there was an absence of colors. The graph demonstrates the numbered wells and the concentration. The fluid goes from 1,000 microliters and is micropipetted gradually until well 12, where the fluid is about 0.5 microliters. This graph demonstrates that Panda 2 had a concentration of 291 microliters, whereas Panda 1 had a concentration of 9.11, showing that Panda 2 had a greater concentration of the hormone. Thanks for watching Harmony Public Schools.